Hello everyone, my name is Ayse Demirkan. I'm affiliated at the University of Groningen from the Netherlands and University of Surrey from the UK. This is a pre-recorded lecture. In the second lecture of on-demand sessions, Introduction to the Statistical Analysis of Genome-wide Association Studies, I will be talking about considerations for genotyping, quality control, and imputation in genome-wide association studies, GWAS. So here you see an overview of the lecture. We will shortly go over genotyping platforms and options, quality control. Then I will talk about definition and purpose of imputation and how it is done. And this is going to include reference data, tools, analysis of imputed data, imputation accuracy, and QC. What we call as genotyping is the process of determining differences in the genetic makeup, hence the genotype of an individual, by examining the individual's DNA sequence. Of course, the technology used for genotyping depends on the structural properties of the genetic variation, whether it is a single nucleotide polymorphism or a copy number variation or other structural variations. It also depends on the project rationale or scientific question and your budget mainly. And related to that, of course, how many SNPs you want to genotype if it is a genome-wide association study, and number of individuals you would like to include depending on your study design. You will also be limited with your DNA sample quality and quantity. So here on this slide, you see the most common approach used for genotyping SNPs, and depending on your study, you will be most likely using one of these. What are those? Illumina Affymetrix arrays. So on the y-axis, you see the number of SNPs that are easily captured by the arrays, and on the x-axis, you see the number of individuals. And then what do we have? We have PCR, RFLP, Sequenome, pyrosequencing, and Fluidigm platforms, and TACMAN. For instance, one of the best examples are the Illumina arrays for whole genome scans. Whole genome genotyping by these arrays provide an overview of the entire genome and enable genome-wide discoveries and associations. So using high throughput next generation sequencing and microarray technologies, you can obtain a deeper understanding of the genome because you are covering a very wide proportion of the genome. So you can use one of their selection of this Illumina or Affymetrix arrays which you think may be suitable for your study. There are many options. As for Illumina, there are genome-wide genotyping arrays for 18 species at the moment. So number of markers on each array change by product. For human, up to 4 million markers per sample are possible now. And then there is an Infinium low-cost screening array. So for this one, for example, includes 600,000 markers on it. You can use start from 200 nanogram genomic DNA. And what you can also do, you can add some custom marker panels. There's an add-on capacity up to 50K markers. And then there is this Omni family of Illumina arrays. Here you see a simple description of their coverage and the inclusion of genetic markers in relation to their minor allele frequencies. So these express chips on the left include only common variation with minor allele frequencies higher than 5%. Some include CMVs and some include SNPs with lower minor allele frequencies. So which one to choose among those will depend on your research question and population you want to screen. For instance, are you looking for a rare or common variation in terms of SNPs? Are you looking for CNVs? Are you working with a rare or common disease? And what is your sample size and your budget? Now, I listed some websites here. Please take 10-20 minutes to check on the technologies mentioned in the first section using these websites. Now let's talk about genotyping quality control, QC. You designed your study, you chose a proper array platform service you used a service from your institute. So one critical initial step from chemically induced intensity signals and data analysis is a transfer and QC of genotypes determined towards your computer. This critical step is called genotype calling. So genotype calling algorithms are always implemented in the proprietary software accompanying the genotyping platform you choose, so you don't need to invent them yourself. So a typical calling software uses a sort of mathematical clustering algorithm to analyze the raw intensity data, and it estimates the probability that their genotype is one of the AA, AB, or BB for a given individual for a given B allelic marker locus. So one method of checking initial SNP quality is visually inspecting the intensity clustering of a particular SNP in the overall population. And depending on this, one can decide whether a SNP is characterized by a clear signal or not. So here on the left of this figure, you see a clear intensity clustering of a SNP in the population. So you see that the common variant is depicted by red on the left. There is some heterozygous in the middle depicted by purple, and the homozygous people for the less common allele are depicted as blue. And the table on the right, it shows the raw values that this plot is figured from. So here the plot shows a tight clustering of genotypes, and there is not much noise in the measurement of this SNP. Following that, there are imputed the important data QC steps. One of them is to work on replicates. So for inspecting plating issues and by looking at genotype concordance, this would be a good thing to do, to include the same DNA sample on different batches of experiments. And then there are Mendelian errors to control for instance, transmission and the inconsistencies. For example, SNPs with more than 10% Mendel error rate can be excluded. This would be based on the number of trios that you would include in your experiment. Unfortunately, this option obviously is only available for family-based and trio designs only. Another QC measure we use is SNP call rate. This is basically the missing genotype rate, 1 minus the missing genotype rate per SNP. So this can depend on the quality of the assay, and this is generally between 95% and 99%. This is a very standard thing to include in your QC. Another thing is the Hardy-Weinberg equilibrium deviance of your SNP. So this is another method for checking the quality and exclusion of your SNPs. This will be explained in the next slide. Another one is the sample call rate. So this is a sample based QC method. This is a good indication of sample success. So different platforms have different thresholds, but this will be mainly determined by your initial DNA quality. And it will somehow be in relation to with SNP call rate. So once you do SNP call rate, you could do sample call rate, and you may want to repeat SNP call rate depending on that. And another thing to do is sample gender check. For this quality measure, you need X chromosome information to calculate this. And you may want to add this as an additional sanity check in your data to make sure that there is a perfect overlap with your phenotype files in terms of sex. Another important one is sample heterozygosity. This is to check for sample outliers. For example, samples with more heterozygosity than expected can be an indication of contamination in your samples. And you also want to do something on top of all of that. You need to check samples cryptic relatedness and unexpected twinning, and whether there is actually a relatedness and structure in the data. But this will be more covered in the lecture of Reddick-Magee. So let's talk about Hardy-Weinberg equilibrium shortly. So as occurrence of two alleles of a SNP in the same individual are two independent events, the distribution of the genotypes across individuals should be more or less in equilibrium with the frequencies of the alleles of a biallelic SNP. So this is only possible in ideal conditions, of course, which would be random mating, no selection, equal survival, no migration, no mutation and selection based on mutation, no inbreeding, and large population size. So under these conditions above, deviations from Hardy-Weinberg equilibrium is an indication of genotyping calling problems, and a commonly used threshold for genotypes variance is a p-value of Hardy-Weinberg equilibrium that is less than 10 to the minus 5 is an indication of a deviation from Hardy-Weinberg equilibrium, and you may want to take a look at these SNPs, or you may want to exclude them from your data set. Another important thing to always consider is genome builds and alignments. So the characterization of the human genome is an ongoing effort. And a genome build tells us the positions of the SNPs in the genome, on the genome. So the latest build is called build 38, but the most commonly used one at the moment is still build 37. For instance, the HapMap was released on build 35 and build 36. So you need to be aware of issues relating to merging and meta-analyzing data from different genome builds. Also, for when preparing your data for imputation, this is very important because you need to make sure that your data is coded according to the same genome build between the target set and the reference data set. So there are tools for that one. They are called liftover tools. For instance, there is one from Oxford that we use for purpose, and I provide the link to that here. So all of these QC steps I shortly went over here are pretty standard, and there are a couple of widely used tools. One very commonly used tool that we also use for data storage, analysis, and QC is called PLINK. Here on this slide, I made snapshot of some of the PLINK options that I also covered during the lecture. And these functions are implemented in the PLINK software, and you can use it for the QC of your genetic data. So first thing, to be able to use PLINK, to obviously install PLINK, and you will need to read your genotype call data in PLINK in the form of map or pad files. And then you can perform QC at the SNP level, remove or extract SNPs, and you can perform QC at the sample level. You can remove or extract individuals. And under the summary statistics option here, there are functions listed to check for call rate, missingness, Hardy-Weinberg equilibrium, allele frequencies, Mendel errors. You can also perform sex checks. What PLINK can also do is to extract genetic principal components and identify cryptically related individuals or twinnings in the data, and the genetic structure of the data, and which you can then use to determine ethnic outliers in your dataset. I will not talk about this because this is a part of the lecture of Reddick Maggie of the next session. Now, here I put two websites here. One of them is for PLINK and how to use PLINK for QC, and the other one is for Beat Studio, which I mentioned is one of the algorithms that you could use for genotype calling. So now take 10-20 minutes to have a look at these websites and try to grasp what you can do with them. Now let's talk about imputation. Why do we need imputation? We need imputation to address missingness in the genetic data. This is all about missing values in the genetic data. Where do the missing values come from? So during the QC, we already set some values to missing, right? And also during genotype calling, you could set some data points to missing. But actually, most of the missing values come from the initial targeted coverage of the genotyping chips and platforms we used. So remember that there are many types of arrays, some of more dense, less dense. There are arrays made specifically for oncological studies, like oncoarrays. There is a metabot chip that is designed for metabolic disease especially, and there are arrays focusing on mainly SNPs with high minor allele frequency, or there are arrays focusing on CNVs. But even the dense SNP arrays do not cover all of the genetic variation. They cover much less than you would imagine. And in addition to that, SNPs included in one array may not be included in the other one. And for many variable positions on the genome, we do not have matching information across genotype set of individuals. For instance, look what I try to depict here. Think of three individuals. First two are typed on the array X, and the third one is typed on array Y. So hence they have different missing data points, and when you try to pool their data for a pooled analysis or to be used in meta-analysis, you're going to have even more missingness in this data because of the non-overlapping positions, and you will not be able to replicate findings from one of data set and in the other one. So additionally, we will be analyzing only half of the genetic variation, and we may miss causal variance in the analysis. This is because of all these reasons we use genetic data imputation. So what do we do in principle? In principle, it means estimating the most likely genotypes in an individual at the missing positions by looking at the correlated SNP values from a more complete data set, and based on that, writing over the missing values in the target data set. So how does it work? First of all, we need a data set where dense genotypes are directly measured. This can be a dense SNP array, or it could be a set of sequence individuals. This we call a reference panel. Then we use an imputation software or service, and by looking at the correlation structure of the densely typed or sequenced SNPs, we estimate them in the target data set. So at the end, these are probabilities, and we end up with dosage information for alleles or genotypes rather than hard genotypes calls. And this dosage information, which accounts for the ambiguity in the estimation, is then included in the genome-wide association study analysis. So to sum up, the purpose of imputation is to increase power, because obviously the reference panel is more likely to contain the causal variant than a less dense GWAS array. To improve fine mapping, because imputation provides a higher resolution overview of an association signal across a locus. And then to enable meta-analysis, because imputation is going to allow GWAS typed with different arrays to be combined up to variance in the reference panel. Going over the historical milestones in terms of imputation also summarizes the theoretical and technological advancements in human genetic data imputation. So one important advance in all of these was the generation of reference panels. So the first reference panel was HapMap, and the HapMap 2 was the most commonly used release of HapMap. It consists of a limited sample of individuals from diverse genetic backgrounds: 60 Yoruba Indians, 90 Han Chinese and Japanese, and 6 individuals that were Utah residents descending of European ethnic origin. Now it sounds funny to think that we imputed thousands of people based on the genetic material of 60 Europe residents only, talking about the Europeans, but actually this yielded a lot of success, and actually this is what we had only for a long time. So we could only impute up to 3 million SNPs with HapMap at the time. And then came the 1000 genome reference panel, which included at the end 2500 individuals from multiple ethnic groups. And later on, and currently the most widely used reference panel is the Panel of Haplotype Reference Consortium, HRC we call shortly. This is a combined set of whole genome and exome sequence data for more than 30,000 individuals and yields 39 million SNPs after imputation. Of course, this is going to depend on the scaffold that you use for imputation as well. Many of these SNPs will not be imputed with a good quality, but in the ideal conditions you can go up to 39 million. And finally, we now have a reference panel from the Transomics for Precision Medicine TopMed program, and this consists of almost 100k deeply sequenced human genomes, and it can yield up to 308 genetic variants to be identified. One technical milestone needs mentioning was prephasing of haplotypes. So genetic imputation is a highly computationally intensive process because of the probabilistic framework and high rate of missing data that we are trying to deal with. One of the major milestones is to reduce the computational burden was introduction of prephasing. So this idea involves a two-step imputation process. So there is one initial step of prephasing, which is actually haplotype estimation of the GWAS genotypes, and a subsequent step of imputation into the estimated study haplotypes. So this reduces complexity of the imputation process and speeds it up. The current version of all imputation software can deal with the prephasing approach. And what is very important is the choice of reference panel. So it is shown that making use of the all ancestries reference panels rather than ethnic specific reference panel improves imputation accuracy for rare variants in any population. Formatted reference panels for Impute and Minimac can be downloaded from the software websites, and it's very important to make sure that genotype scaffold and reference panels are aligned to the same build of the human genome. I will get back to that later as well. So another and very important and current technological advancement that makes our lives easier is the imputation services. These are freely available services such as the Michigan and Sanger imputation services. You can simply format and upload your data in a secure way to this server and get the data imputed and phase genotypes back in a few days. And this depends on the speed and how busy the server is, and depending on the sample size you are trying to impute, of course. So in parallel to Michigan imputation server, there is also a Sanger Institute has a similar service. In this service also you can upload your data in a VCF format and optionally perform prephasing using Beagle or Shapley software. And current reference panels in the Sanger imputation server includes HRC, UK10K, and 1000 genomes. As I said, there is also a server dedicated to the TopMed. This is all very self-explanatory. This is how the Sanger imputation server would like you to prepare the data. So there is a whole bunch of instructions there that you would like to use. So the use of these services comes with instruction and manuals. So feel free to make an account there and run some test data sets in there. You will need to QC and format the data as required in the instructions. You will need to match the coordinates and the reference allele of the genome builds and prepare one file for each chromosome. This is for Sanger imputation server. And another important thing in terms of imputation is, of course, the speed. So increasing reference panel size improves accuracy of markers with low minor allele frequencies, but this poses ever-increasing computational challenges for imputation methods. So recently a new imputation software, Impute 5, was introduced from the same group. So it does memory-efficient imputation by selecting haplotypes using the positional Burrows-Wheeler transform. So using HRC reference panel, the developers of the software showed that Impute 5 is up to 30 times faster than Minimac 4 and up to 3 times faster than Beagle 5.1 and uses less memory than both of these methods. So, using all the mentioned considerations up until now, you can build an in silico framework similar to this one. So, you can use, for instance, PLINK functions for the first two steps of genetic data QC. Then you can check chip information and strands issues using Rainer tools. And if needed, you can update your genome build by using liftover tool, and you can then prephase by using Shapeit, and finally impute it in-house or using one of the servers are mentioned. So links to this software are given here. Now, take time, probably hours, to explore these three imputation services of Sanger, Michigan, and TopMed. You will be asked to make an account, and perhaps it will need to be improved, so take your time to do so. And the next topic is imputation related QC. So there are two QC steps around imputation. One is pre-imputation QC. So we have already discussed standard QC after genotyping, and on top of that you may want to exclude SNPs with less than 1% minor allele frequency. And post-imputation quality is assessed by information measures, which is in some value in the range of 0 to 1. And it is typical to filter SNPs by this value less than 0.8 for a strict filtering, or less than 0.4. And in the impute software, this is called InfoScore. And in the Minimac imputation software, this is called R-square per SNP. So it's important to check quality of type SNPs in the scaffold in the region, also by visual inspection of cluster plots. And you may also want to produce quality plots per chromosome, varying by minor allele frequency, strata, and position on the chromosomes. For instance, this is an example figure. This shows a typical relationship between minor allele frequency and imputation quality. So on the y-axis you see the imputation accuracy as determined by imputation quality as by R-square or InfoScore from different softwares, and on the x-axis you see the minor allele frequency. You see that the accuracy is top when the minor allele frequency is high, when the allele is common and more common, and then the accuracy goes lower where the minor allele frequency goes lower as well. You still have some SNPs with well imputed SNPs among the rare ones as well, but most of the low quality SNPs are going to come from low minor allele frequency SNPs. So keep in mind that when you filter by imputation quality, you will be filtering out a lot of rare SNPs as well. So what are the factors affecting imputation quality? So at the genome-wide level, the number of individuals imputed has something to do with it. For this reason, we merge scaffold data sets before imputation if we are going to impute more than one. So the more the merrier. And the second factor is the reference panel. The choice of the reference panel as well, as the whole idea is to use the correlation between SNPs across different populations, and this may be different from population to population. You want to go for a large multi-ethnic panel if you're not able to go for a large ethnic specific panel. And finally, at the SNP level, the lower the minor allele frequency, the lower the quality of the imputation is going to be. And how to analyze the imputed data? So for each individual, imputation provides probability distribution of possible genotypes for each untyped variant. These probabilities can be converted into best guess genotypes, but this is not something really generally recommended, as it increases false positives and it reduces power. But also you want to filter your best guess genotypes. You want to put a strict filtering on the best guess genotypes, and this would result more NAs in your data set. So it's better to convert the probabilities to expected allele counts and analyze by taking the uncertainty in the imputation into account. That's really important. To do that, you need to match the data formats to the software. Not all software uses all types of data, and you may need to do data conversions. Software called EPAX, SNP-test2, and PLINK to support dosage information. And you need to check the lecture from Medic Magi for analysis of genome-wide data. So this is the last slide of this lecture. So the take-home message is that as we are dealing with hypothesis-free approaches here, unfortunately it all comes down to the bittersweet money and resources we have. So you need to think what is the best and most cost-effective way of getting genetics done in large sample size. And the answer is combining a dense genome scan array with imputation, as the reference panels are free at the moment and cost of arrays are going lower as well. But you really need to think, as the in silico part of doing so also will cost staff and computational resources to some level. And what else should you consider? So in comparison, you want to know, depending on your research question, of course, whether there is a better array for you, or perhaps an onco array or a metabot chip if you are gonna conduct your research in a very restricted field. And the most importantly, what are the future uses of this data? Because obviously you don't want to build something that you're going to use only a couple of years and finalize the research on that. You ideally want to invest in big data. So are you going to invest in population-based cohort or disease-based cohort? Is it going to be a short-term project, or is it going to be a follow-up study that's going to be likely built up and extended throughout the years and by inclusion of new phenotypes? So, and finally, who do you want to collaborate with, which consortia, which disease? So I hope this lecture will be useful for your research and future studies. And for the people who are interested and to have a better and in-depth understanding of imputation, every year 2 times we have a GWAS course organized by University of Surrey in collaboration with Imperial College and University of Tartu from Estonia. This includes a hands-on workshop as well as theoretical lectures where we teach these concepts and methods in more detail. So the last one was in 5 to 10 July 2021, and for information there is an email address you can connect to. Thank you very much, and have a nice conference of the remaining time.